This video will cover the following objective from the reproductive system. Describe the location, structure, and function of the testes. Define spermatogenesis. Describe the location and functions of the spermatogonia, spermatocytes, spermatids, spermatozoa, Sertoli cells, and Leydig cells, or interstitial cells of Leydig. The testes are found within the scrotum. The scrotum is a sac of skin located just inferior to the pelvic cavity. The scrotum contains the testes outside of the pelvic cavity in order to maintain the optimal temperature for spermatogenesis, the formation of sperm. The scrotum has a layer of smooth muscle in the dermis of the scrotum, known as the dartos muscle, that can contract involuntarily in order to produce the wrinkled appearance of the scrotum that helps to draw the testes closer to the pelvic cavity, helping to increase the temperature inside of the testes. Similarly, there's a skeletal muscle known as the cremaster muscle that can contract to pull the testes closer to the pelvic cavity to warm the testes. And both the cremaster and the dartos can relax in order to move the testes further from the pelvic cavity to help cool down the testes. The cremaster muscle is found within a bundle of connective tissue known as the spermatic cord. The other structures that are deep to the cremaster muscle within the spermatic cord are the testicular artery, which is the male gonadal artery carrying blood into the testes. There's also the testicular vein, or the pampiniform plexus of testicular veins that carry blood out of the testes, and the testicular artery and the plexus of testicular veins have a countercurrent flow mechanism that helps to conserve the heat within the blood and prevent that heat from flowing into the testes. The heat is transferred directly from the testicular artery into the plexus of testicular veins as blood is flowing in the opposite direction through those vessels. Another structure found within the spermatic cord is the ductus deferens, or vas deferens, the tube that will transport the sperm from the scrotum into the pelvic cavity to the ejaculatory duct in the prostate gland. The testes contain small, tightly coiled tubes known as the seminiferous tubules that are responsible for sperm production. The cells that form the seminiferous tubule include spermatogonia and spermatocytes that are involved in spermatogenesis. The spermatogonia are found in the most superficial layer of each seminiferous tubule, and as they divide, producing primary spermatocytes, the primary spermatocytes are pushed deeper towards the lumen and enter meiosis. Each primary spermatocyte will produce 4 spermatids in 2 rounds of meiotic cell division. As each of these daughter cells is produced, they will be pushed further towards the lumen so that the spermatids are found adjacent to the lumen and are released into the lumen as they enter the process of spermiogenesis, where a spermatid will mature into a spermatozoa, or mature sperm cell, that will develop a tail to propel itself through the female reproductive tract to find the developing oocyte and initiate the process of fertilization. The other type of cell that's found in a seminiferous tubule is known as a Sertoli cell. The Sertoli cells are surrounding the spermatocytes within the seminiferous tubule, and They function to help support the process of spermatogenesis. Sertoli cells produce a hormone known as inhibin. Testosterone is produced by another cell that's found just outside of the seminiferous tubule, known as the interstitial cell of Leydig, or just Leydig cell. The interstitial cell of Leydig produces testosterone in response to another hormone that comes from the anterior pituitary called luteinizing hormone. Testosterone will have a function inside of the seminiferous tubules to stimulate spermatogenesis, and testosterone will also have effects all through the body, stimulating masculinization, like increased facial hair growth or growth of the larynx to produce the lower pitch voice in men, or stimulating the growth of the skeletal muscle. Here we see the histology of the testes at low magnification, where you can see there are numerous seminiferous tubules responsible for spermatogenesis, where the spermatogonia will divide by mitosis to produce primary spermatocytes that enter into meiosis to produce the spermatids. The spermatids then enter into the lumen of the seminiferous tubule and go through spermiogenesis to form the mature sperm cells known as spermatozoa. The outer layer of the testes is fibrous connective tissue known as the tunica albuginea. Here we can see regions where there are interstitial cells that produce testosterone. We can see the spermatocytes that are going through meiosis to produce spermatids that are released into the lumen to form the sperm or spermatozoa. The process of spermatogenesis is regulated by the endocrine system. The hypothalamus produces a releasing hormone known as gonadotropin releasing hormone, or GnRH. GnRH is released into the hypophyseal portal, and it travels in the blood into the anterior pituitary, where it will stimulate cells in the anterior pituitary to release FSH and LH. Luteinizing hormone will then bind to receptors on the surface of the interstitial cells of Leydig and stimulate the production of testosterone. Testosterone will stimulate the process of spermatogenesis, but testosterone will also travel through the blood to have masculinizing effects, and in the hypothalamus and anterior pituitary gland, testosterone will have a negative feedback function to decrease the production of GnRH and LH. Follicle stimulating hormone produced by the anterior pituitary will bind to the receptors on the Sertoli cells in the seminiferous tubules. Then the Sertoli cells will respond by producing androgen binding protein and a hormone called inhibin. Androgen binding protein functions to bind to testosterone inside of the seminiferous tubule, helping to increase the local concentration of testosterone in the seminiferous tubule, helping to promote the process of spermatogenesis. Inhibin functions as a negative feedback hormone to decrease the production of follicle stimulating hormone from the anterior pituitary. Both inhibin and testosterone have negative feedback functions, leading to a decreased production of FSH and LH as a homeostatic control mechanism, maintaining a stable concentration of FSH and LH in the blood.